Okay, hey, my name is Philipp. I want to quickly walk you through this poster about a project where we looked at the response of acute myeloid leukemia cells to chemotherapy. So our disease of interest, acute myeloid leukemia, is a type of blood cancer that affects the myeloid lineage in the bone marrow. And it is usually treated by chemotherapy, but one major problem about AML is that patients very often relapse after chemotherapy. This can in part be contributed to a very high heterogeneity in the tumor. So, for example, within one patient, you can have clonal evolution in a sense that a certain subclone survives chemotherapy treatment and then expands at relapse. So our question here for this project was, what are the molecular changes in the tumor cells if they undergo repeated chemotherapy and relapse? For this we choose patient-derived xenografts as our experimental model, where we transplant primary tumor cells of a human patient into immunodeficient mice, and then we can treat these mice with chemotherapy, monitor the tumor burden by in vivo imaging, and finally re-isolate cells for further analysis. So this is the experimental setup that we choose. So we used primary tumor cells of a female patient that were isolated after the first relapse. And then we transplanted these cells into mice, amplified them and transformed them so that they express luciferase, so that we can monitor the tumor burden as seen in these bioimaging pictures at the bottom. These cells were then re-isolated and transplanted into different treatment groups of mice, where the mice were then either left untreated, as in treatment group 1, or received 1, 2, or even 3 blocks of chemotherapy treatment in a time span of up to 55 weeks. After this, the mice were sacrificed either directly after chemotherapy treatment, so in a depleted tumor state, or after the tumor burden had increased again. We then used a range of different methods to characterize the transcriptomic, but also genomic, epigenomic, and proteomic features of these cells. And here you can see our first results just of the bioluminescent imaging analysis, where we observed that in this experiment, the AML-PDX cells did not develop chemoresistance. So in all mice that received chemotherapy, we saw first a decline in the tumor burden and then a subsequent relapse. However, the growth rate of the tumor cells slowed down after repeated chemotherapy, while the decline rate did not change significantly. We next used whole genome sequencing data from the patient at diagnosis, relapse and relapse 2, together with 5 PDX samples to infer the clonal phylogeny. In the patients, we observed one major subclone per sample that derives in a linear fashion from the founder clone. In the PDX lines, we found 2 major subclones, one with an NRAS mutation, the other one with a KRAS mutation. So that's where these names come from. We then went on to characterize the substitutions that define the different subclones in more detail using signature analysis with YAPSA. For the founder clone, we found that most mutations are associated with signature 1, which probably just reflects endogenous mutational processes. In contrast, the substitutions for clones that arise after chemotherapy, so especially the relapse 2, NRAS and KRAS clone, showed an excess of signature 18, which is known to be associated with reactive oxygen species. So this shows that the exposure to chemotherapeutic agents could lead to a non-endogenous mutational regime that results in these similar patterns in both the patient and the PDX lines. So to estimate the clonal composition in a greater number of PDX samples, we performed whole exome and genome resequencing for many more PDX samples. Especially after the first block of treatment, the NRAS subclone increased in frequency. Based on the estimation of growth and decline rates, this could be a result of the higher sensitivity of the KRAS clone to this first treatment block with cytarabine and daunorubicin, which is slightly different from the subsequent treatment blocks. Finally, we performed single-cell RNA sequencing on a range of PDX samples to assess the transcriptional heterogeneity and changes along this relapse timeline. Within the main clusters, we observed cycling cells, stem cell-like cells, but also cells that appear to be more differentiated. We classified the cells with a reference of cell types that were present in the healthy bone marrow, and this revealed that in our experiment, the AML blasts show a similarity to a range of different cell types along the myeloid lineage, ranging from HSCs to monocyte-like cells. Interestingly, in the depleted stages, so where mice were sacrificed shortly after chemotherapy treatment, the fraction of more differentiated GMP or monocyte-like cells was higher, but the fraction of HSC, so hematopoietic stem cell-like cells, decreased. Similarly, we saw in a differential expression analysis an upregulation of immune signaling pathways and a downregulation of genes associated with translation in these cells, from the depleted stages. Okay, so in conclusion, we observed a slowdown in the growth rates as a result of repeated chemotherapy treatment, which is associated with a depletion of stem cell-like features and a more differentiated transcriptomic phenotype. So with that, I'm at the end, and I'm happy to answer further questions over in the poster booth.